Welcome to our monthly Fireside Chat. I'm Doug Montgomery, your instructor for the Design of Experiments MOOC. I host this Fireside Chat session on a monthly basis. I always have a guest speaker, and we explore important topics and applications of design of experiments. We always set aside some time for questions and answers, and so whether you're taking the Coursera courses or applying experimental design methods in your work, or simply curious to learn more about this really intriguing field, this is a great opportunity for you to ask questions and engage in discussion. If you can't join us live, we always record these fireside chats and post them in our LinkedIn page, which is linked to our YouTube channel. And we will share the link to this for you in the meeting chat. If you're a Coursera participant, these recordings are also available in your course under the Resources tab. Quick reminder, please continue to keep your microphones muted and your cameras off so that we don't have any miscellaneous noises during the session. Things like barking dogs and crying children. We don't, it's that's part of life, but hopefully not part of the fireside chat. And if you want to submit a question to either me or our guests, please do this using the chat function on the Zoom menu. You don't need to wait until the Q&A starts to submit your question. You can start putting that in at any point during the session. And as I say, your question can relate to really any aspect of experimental design. The talk that our guests are going to give, or maybe an application that you're involved in and you'd like to get some advice from us about how to proceed. If your question doesn't get answered during this time period, we will respond to it in Coursera for those people that are enrolled. Okay. I have two guests today. We're lucky. We've got two really great guests, Brickey and Peili Wang. They're both statisticians at Pfizer, which is, of course, well known in the biopharmaceutical industry. They're one of the world's top companies. Their jobs are to provide statistic support to scientists to ensure the quality and consistency of pharmaceutical products and during late stage development and manufacturing. Peili received her PhD in statistics from the University of Florida. Katie holds a master's degree in statistics from Virginia Tech, my old alma mater, and go Hokies. and I am really pleased to have both of them here today. So I'm going to turn the show over to them.
Thanks so much, Doug. I'll share our presentation. Very good. Can you see it okay?
Yes.
Okay, great. So we'll go ahead and get started.
I've got it.
Okay, perfect. Thank you. Thank you all for joining us today, and thanks, Doug, for inviting us to your fireside chat. Peili and I are excited to share about our work as non-clinical statisticians in the pharmaceutical industry, and specifically about how we use design of experiments in our work. The title of our presentation today is A Case Study on Design of Experiments in the Pharmaceutical Industry, Leveraging an Experimental Database to Inform an OMARS Designed Experiment. So as Doug mentioned, Peili and I are both non-clinical statisticians at Pfizer. Peili supports research and development for biotherapeutics, and I support manufacturing and quality for small molecules. We both commonly use design of experiments in our work. And in our presentation today, I will start by providing an overview of some of the work we do at Pfizer as non-clinical statisticians. And then I'll hand it over to Peili, who will describe an interesting application of DOE that we're both currently working on together. So as Doug mentioned, Pfizer is a large pharmaceutical company, and this slide provides an overview of Pfizer's history. Pfizer was founded in 1849 by two cousins. One was a chemist and one was a confectioner. And these cousins produced an almond toffee flavored medicine shaped like a candy for intestinal worms. Today, 176 years later, Pfizer is a world-leading pharmaceutical company with over 80,000 employees and a portfolio of hundreds of medicines and vaccines, which are distributed to patients globally. Now, some of you may know that the history of designed experiments lies often outside the field of statistics. Early pioneers in DOE were not necessarily statisticians by training, they were scientists, chemists, and biologists, maybe even a confectioner, who were conducting experiments and wanted to apply scientific rigor to the design and analysis of these experiments. And the same is really true today of the scientists who work at Pfizer, who Peili and I support. To develop and deliver breakthroughs that change patients' lives, we need to collect the right data and we need to understand what conclusions are supported by that data. So Pfizer's purpose is to develop and deliver breakthroughs that change patients' lives. And we do this through a relentless commitment to innovation, tackling some of the world's greatest health challenges. As non-clinical statisticians, Peili and I are one piece of Pfizer's innovation pipeline. Innovation requires experimentation, and innovating at light speed requires efficient and effective experimentation. And this is where statisticians like Peili and I provide our expertise to advocate for statistically designed experiments, and then to coach and guide Pfizer scientists on the design and analysis of these experiments. As I mentioned, Pfizer manufactures and sells more than 600 medicines worldwide. Some of them I'm sure you've heard of, such as the coronavirus vaccine Comirnaty, or Lipitor, which is used to lower cholesterol. Now, developing a new drug requires both clinical and non-clinical activities. In the clinic, patients are enrolled in clinical trials to study the efficacy of a new drug. Patients are assigned to control groups where they would receive a placebo dose, or to treatment groups where they receive a dose of the medication being trialed. And a clinical statistician supports the design and analysis of these clinical trials, which determines the efficacy of new drugs to treat different diseases. Now, the medicines that are used in clinical trials and then subsequently sold worldwide to patients, they have to be made, right? They have to be manufactured from raw materials into the final drug product, like the vaccine or the tablet that you might take. And this is where Peili and I come in as non-clinical statisticians. Specifically, Peili and I are both CMC statisticians, and CMC stands for Chemistry, Manufacturing, and Controls. What is CMC? After a lead compound is discovered, a supply of the compound is required for testing to establish the compound's safety and efficacy. The manufacturing processes and analytical methods evolve to support the scale and quality required to supply the drug. So from early clinical trials through late stage, and then commercial selling of the product. And statistics plays a significant role in every step. Some of the key statistical methods we use include design and analysis of experiments, assay development and validation, predictive modeling, and statistical quality control. And so I'll provide some examples for each of these areas. Now, of course, we're going to talk more about design of experiments in a moment, but at a high level, DOE provides a systematic approach for collecting data to understand how a drug manufacturing process operates. So Peili is going to describe a bioprocess for drug development, which uses living systems like cells, enzymes, or microorganisms to create biological drugs. These are very complex processes. So a statistically designed experiment might study the impact of factors like pH, temperature, and time on cell growth and cell viability. Another area we support is assay development and validation. The assay development and validation is the process of designing and optimizing an analytical test or an assay to accurately measure a compound. We need precise and accurate analytical methods to measure the quality attributes in a designed experiment. Developing analytical methods also often involves statistically designed experiments. We need to determine what instrument parameters create a reliable and reproducible analytical method. So for example, we might study the impact of column temperature or flow rate in order to optimize an HPLC method. A third area is predictive modeling. Now, predictive modeling has many applications, and of course, one is that we develop empirical models based on the data collected in a statistically designed experiment. So this is one example of predictive modeling. Another are shelf life analyses. Shelf life analyses are an example of predictive modeling where we study how storage conditions impact critical quality attributes over time. We want medicines that are stable over their shelf life, and so we design shelf life studies and conduct analyses to collect stability data, and then develop a predictive model to quantify if there is a change over time. And finally, statistical quality control techniques are vitally important for monitoring and improving manufacturing processes. We use tools like control charts and capability indices to understand variability. For example, when Pfizer manufactures a drug, we monitor key quality characteristics like yield for every batch produced. And we plot these results on a control chart to visualize typical and atypical trends. So for today, let's talk more about design of experiments. In the pharmaceutical industry, we're really lucky that the structured approach of statistically designed experiments is supported by regulatory bodies worldwide. So for example, here are 3 guidances from ICH. ICH is an international council harmonizing technical requirements for pharmaceuticals across regulatory authorities in different countries. So like the FDA in the US or the EMA in Europe. ICH Q2R2 describes the validation of analytical procedures. ICH Q8R2 describes pharmaceutical development, and ICH Q1 describes stability testing of new drug substances and drug products. These are guidance documents. They're not prescriptive, but their direct mention of statistically designed experiments encourages drug developers like Pfizer to employ these methods. So for example, in ICH Q8R2 on pharmaceutical development, the introduction states, a more systematic approach to development, also defined as quality by design, can include incorporation of prior knowledge, results of studies using design of experiments, use of quality risk management, and use of knowledge management. Such a systematic approach can enhance achieving the desired quality of the product and help regulators to better understand a company's strategy. So in this way, the use of statistically designed experiments is interwoven into the pharmaceutical development process and clearly supported by regulatory authorities who want to see a systematic approach used. DOE is not nice to have, but a key tool in our tool belts. And as non-clinical statisticians, we support these activities. So now I'll talk a little bit more specifically about designed experiments for late-stage drug development with some topics that you're probably learning about in this course. Experiments can be designed to address different objectives. So for example, in screening studies, we ask what factors significantly impact the response. In screening studies, we want to separate the vital few factors from the trivial many. And we might study a relatively large number of factors in a relatively small number of runs. Common screening designs include fractional factorial designs, definitive screening designs, and supersaturated designs. In process characterization studies, we want to understand what is the impact of process parameters on critical quality attributes, and what operating ranges produce a product that meets all acceptance criteria. In characterization studies, we aim to study these vital few factors in more detail to quantify their functional relationship to the response variables. Common characterization experiments might include fractional factorial designs and optimal designs. In optimization studies, we might build on a process characterization study to determine what are the best operating conditions. Determining the best operating conditions requires an accurate empirical model which sufficiently represents the underlying process. One common type of optimization study is a response surface experiment. Finally, in robustness studies, we define within what region can the process operate without significantly impacting critical quality attributes. Robustness studies are an important tool for non-clinical statisticians, though they can be a bit funny to think about. So in contrast to screening, process characterization, and optimization studies, the goal of a robustness study is to find a region of no significant change. So we design experiments of different sizes and different structures according to what objective the study needs to meet. So my comments so far were intended to provide a broad general overview of our role as non-clinical statisticians, and specifically how we support experimentation in the pharmaceutical industry. And now I'm going to hand it over to Peili to discuss a specific case study that we're working on together, which involves leveraging an experimental database to inform an OMARS designed experiment. Peili, take it away.
Thank you. Thanks for Katie's introduction about our CMC work. The focus of my work is in biotherapeutics. I'm going to give background about this topic first, and then introduce the topic. Next. This is a general bioprocess workflow for producing drug substance. Top part, cell culture, is about growing the cell to produce the protein we want. This stage involves progressive culture volume expansion and will produce the target protein under target condition of process parameters such as temperature, pH in bioreactor. Then we need purification steps to remove the host cell and the impurities, the things we don't want in the drug substance, through centrifugation, filtration, and other chromatography steps. For some key steps in this process, we work with scientists on process characterization studies, PC studies, typically. PC studies are usually done to help understand how process parameters impact quality attributes, as Katie mentioned earlier. We usually do a response surface study. So we have DOE studies from different compounds for each key step. What we saw is if we can put all DOE studies together, it could be good prior knowledge to help prepare design or analysis in the future. In this presentation, I will introduce how we prepare the database to inform some common questions for DOE discussion, and introduce how we leverage DOE database to inform OMARS choice, my topic today. Next. So the goal is turning historical data sets into knowledge. There are two parts in my presentation today. First, about building a DOE database. The challenge is turn data to knowledge by dashboard. Second, show a use case about how we use the knowledge from database to inform design choices. Next. Now the first part about database preparation. Next. In bioprocess, from cell culture to purification, there are some key steps require PC studies. For a key process step, we have DOE studies from different compounds. One compound has one study typically. So we have many studies from different compounds for the step. And for each study, we have input and output. Input means process parameters, factors, X variables. And the output is attributes, response variable, Y variables. And there are a lot of inconsistency from cross-compound studies. Not all compounds study the same attributes. Not all compounds study the same process parameters within the same range. Each attribute variation could be different. Different design resolution, data formatting issue, like name and the unit inconsistency. Next. About how we prepare a database and the dashboard, we use a data scientist workbench called Dataiku to build up the pipeline for data preparation and the dashboard. First, we organize a set of tables for each study, including the data table, factor information, attribute information, selected model information. The figure here gives ideas about how we keep the selected model information, including final selected effects, any transformation, or any sample need to be removed. Next. For compiled studies, we set up keys or identifiers for all variables, all potential inputs and outputs, X and Y, and add it into each study files. And also make the units of data consistency. Some attribute may have two different units, then we have two keys and treat them as two variables. A pipeline is built up by R in Dataiku to compile files and the extra information we are interested, such as coefficient, effect size, R squared from each model. So we have ability to extract or compile useful information from all studies much easier. And the dashboard is built to load the compiled information and summarize them based on needs. Next. There are some frequently asked questions in DOE discussion. Which factors were studied? What were the factors ranges? Which factors are most important? Do we expect to see any interaction or quadratic effect for the factors? Is the data range typical for the attributes? Have we seen this effect in other studies? So in order to help answer those questions, we built up a dashboard including these key components. Study range for all factors, data range for all attributes, frequency about factors and the attributes are studied, relative frequency of statistical significance for main effect or for second order effects, effect size, and co-significant effect. Co-significant effect, this is about how often two effects show up in one model. I will introduce the usage in the use case about OMARS later. Next. This shows how our dashboard looks. First, it allows users to select the scope. User can pick the studies they want to focus, can focus on only newer studies or studies more related with their work, and then can select the interesting attributes or factors also. And for the attributes, a column on the right shows how many times these attributes has been studied. The number is changed based on the selected studies. And based on the selected attribute and studies, we know how many models involved in the consideration. Typically, only one attribute is in one study has one representative model, and it could be a mean-only model. Next. After deciding the scope, users can check things like the study range for each factor across studies on the left. Also can check the heat map to show the impact of X to Y, right. Blue color in this heat map implies this input factor has less impact on the attribute. This visualization is available for things like a number of studies, number of significance, or effect size. Next. For second order effect, take one factor as example. Each factor has one quadratic effect and several interaction terms. We also check how often an effect is selected and can check how they co-significant. The right part heat map shows how often an effect is co-significant with other second order effect and can base on count or proportion. It is complex to organize files across studies, but once files are organized, we can display many different insights we gain from data, turn data to knowledge for future discovery. If scientists have questions about related studies, we can pull data or model results and review them also. This wraps up the database part. Next. Now I'm moving to the use case of this database to inform design choices. Next. We received a request about 24-round study with 6 factors for a process categorization study. Scientists want it to be a single block study, so request for a 24-round study. For PC studies, we usually do second-order response surface. That is usually to estimate interaction and or quadratic effects. A full model for 6-factor study includes 27 terms. It's supersaturated. In the past, we did bigger design. We have used CCD and BBD for multiple compounds in this step. It requires more samples and requires 2 or more blocks to finish the experiments. And this experiment takes a long time. Since we learned the new design, OMARS, which bridging the gap between DSD and the CCD, allow the estimation of partial or complete second-order effects, OMARS became a very interesting candidate. Next. For people who don't know OMARS, OMARS stands for Orthogonal Minimally Aliased Response Surface, proposed by Professor Peter Woods. OMARS designs are designs with 3 levels for each factor. Each main effect is not correlated with any other effect. When seeing an OMARS design correlation metric, the gray square part area of 0 correlation. But the correlation between second-order effect could vary based on design, could have different alias structure, and people can pick one based on the prior knowledge or assumption they can apply. OMARS designs allows the estimation of partial or complete second-order effect. In our case, we know from previous studies, not all 6 factors will have an impact on one attribute. Maybe 4 or less factors have impact, and the different attributes could be impacted by different 4 factors. Overall, 6 factors should cover the main interest, and the 24-round is good for full model of 4 factors. This gives us some confidence to try OMARS with the knowledge from database. Next. About using the database to inform design choices. This is a correlation heat map of a 6-factor OMARS design candidate. Unlike the one in previous page, this one does not have equal information for each factor. We can see this from the quadratic term area easier. The quadratic term of one factor is different from other factors, so we don't have the same information for each of the factors. Next. From our database, this heat map shows how often a quadratic term is selected for each attribute. One factor does not have quadratic term very often for almost all attributes generally. This implies that we can accept one quadratic effect has some confounding with the other effect in design. Next. Because there is not equal information for each factor in OMARS design, some designs could be more unequal, more blue between some factors, more red between other factors. Then, which process parameter, temperature, or pH assigned to factors in the design A, B, C, D, E could matter. It can have different meanings. We are considering using co-significant information from database to inform factor assignment. For a 6-factor design, there are 720 permutations, 720 ways to assign factor. An OMARS design can include some strong correlation pairs like the green arrows point. Yeah. We can, based on historical data, pick the assignment to make highly correlated pairs in the design with low chance of co-significant from database on the right. So we have ideas about the feature of a design on the left side, and we have knowledge from the previous study database on the right. And we try to apply the knowledge from database into design choices. We use these ideas introduced here to prepare our design, and the experiment is done. We are working on analysis, and this wraps up the use case portion. Next. To summarize my part, I have shown the ideas about how we build up our DOE database and build up a dashboard to turn historical data to knowledge. And we also share ideas about how to apply database to inform OMARS design choices and the factor assignment. Although this presentation does not touch on the model selection process by using database, but it is in our future work. We are working on the analysis part and may share about the analysis later. This concludes my part. Thanks for your time. I will turn it over to Katie.
Thanks so much, Peili. This has been a really interesting project and a fun project to work on together. And as Peili said, we are currently, like literally right now this week, receiving data from the analytical testing of the samples from this statistically designed experiment. And so we're working on the modeling analysis, and that's ongoing. So maybe Doug will invite us back sometime to talk about that in the future.
Oh, that'd be great.
But for today, I'll just wrap up with some concluding remarks. And in summary, statistically designed experiments are used in all stages of drug development. In CMC, statistically designed experiments play a key role in developing, characterizing, and optimizing drug manufacturing processes. Non-clinical statisticians provide crucial support to scientists conducting experiments. The final key message I want to leave you with is this: it is fun to be a non-clinical statistician. As a profession, non-clinical statistics has a vibrant future, fueled by greater focus on data-driven decision-making in the era of AI. Our roles include refining traditional statistical approaches while also exploring new techniques, which Peili beautifully described for you today with the novel DOE. Increased automation and use of digital tools accelerating our workflows, which Peili also touched on today with the development of the DOE database to inform our experimentation. And finally, we inform the collection of fair data that is findable, accessible, interoperable, and reusable. This involves collaboration from multiple key players. Digital partners help develop platforms for automating certain tasks and developing tools like the historical database Peili mentioned today. Scientists offer their subject matter expertise to inform the design and analysis of the experiments. And finally, as statisticians, we offer our own subject matter expertise in the design and analysis of experiments. While the tools and techniques we use have advanced quite a bit since the early days of DOE, so that now today we include digital partners to support our work, some principles remain the same. And close collaboration between scientists and statisticians is a key ingredient for a successful experiment. So like I said, it is fun to be a non-clinical statistician. In conclusion, we want to take a minute to thank our Pfizer colleagues who supported this work. We're both very thankful to be part of a strong and vibrant Pfizer team that includes great statisticians, great scientists, and great digital support. We also have a few references listed here, both on the OMARS design that Peili described, as well as two textbooks at the end, or an article and a textbook at the end that describe a little bit more about CMC statistics and statistics for non-clinical statisticians. And with that, that wraps up our prepared remarks today. We want to thank Doug and his great team for organizing this event and inviting us to present, and we can take some questions.
Well, thank you too very much for doing this. I'm really impressed with how you've organized a lot of prior historical information about the process that you're studying and the results of prior experiments. I think organizing that in a formal database type way is a big step forward in the process of planning experiments. And you may know from looking at some of the work that I've done that I've long been interested in the process of planning experiments. And I got interested in that quite extensively working with a guy named David Coleman at Alcoa. In fact, we have a technometrics paper that talks about this. And one of the things that I know about planning experiments is, I always tell my students this, years ago, there was a faculty member at Purdue whose name was Charles Hicks. And he actually wrote a little skinny book on experimental design, but he taught in the math department, and he taught a very popular experimental design course. And he always told his students, and I never said it on his class, but many people have told me this, that he said that if you had 10 weeks to solve a problem, you should spend 8 weeks planning the experiment, 1 week running it, and 1 week analyzing the data. And you know, I've discovered throughout my career how important that is. That advice is just amazingly important. And I think what you've done with systematizing the historical data is really important, because I can't tell you how many times I've sat in meetings with people where we're planning an experiment and it's all anecdotal.
Mm-hmm.
Yeah, you know, like, well, you know, I remember we changed that variable, you know, last year, and you know, and you know, pretty soon we're having an engineering seance, you know, or as I sometimes call it, the engineering version of the Vulcan mind meld, you know.
Yeah.
You know,
And our memories are not so good, right,
you are
Doug? I
absolutely
mean,
right. And sometimes we get into controversies, you know, about,
yeah,
yeah,
yeah. I
well, we
remember
tried.
that differently.
Right. We tried that last month, you know,
Yeah.
and this is just a wonderful way to systematize this whole process. And so I think I really encourage the two of you to write this up.
We are working on that.
You really you
Yeah.
should write this up, and if in fact if you want to send it to the journal that I am a co-editor of, I will push it through the publication process, because I think this is so important that people that are actually involved in designing real experiments need to know something about this approach. It's fantastic. I love it.
Thanks so much. Yeah,
Thank you. Thank you.
we are working on we're going to talk about it more tomorrow, me and Peili, in a meeting. We are
Great.
working on writing it up. You know, it's for those of us in industry that can be a little slow because we have day-to-day responsibilities, and then
Yeah,
we have to
it's
go
not
through
part of your
the
job. I understand.
yeah, but we, you know, it is a good thing, and a lot of great work. Peili has done a lot of work developing the database, and it's so funny too. You know, you mentioned the importance of the design of the experiment, and developing this database in some sense might seem kind of simple, you know, sort of just a historical record, but you're right, it's really not. And the fact that it lives with the statistics team and not, I mean, in theory, the scientists could just keep a good record of everything they've done, right? But the fact that it really lives with the statistics team, I think also helps the scientists like really value early collaboration,
Yep.
right? Versus, I'm
Yep.
sure, I know I have examples, I know Peili does, I'm sure you do too, Doug, of scientists who maybe aren't familiar with DOE and have designed and executed their own experiment and then just try to use statistics to rescue the data they have collected.
Yeah,
And
they
that
want the
just doesn't get you
statistician to
far.
come in and sprinkle some magic dust on
Yeah.
this thing that they've created.
Yeah.
And sadly, in many cases, you know, I've looked at that and said, well, you know, I hate to tell you, but this is dead on arrival. You know?
Yeah, right. Yeah, yeah.
Yeah.
That we, you know,
Yeah.
if the knowledge or the information is not contained in the data you've collected, we
Yep.
cannot, you know, force it out.
Yep. You can't sprinkle the magic dust on it and make the answer appear.
Appear.
Well,
Yeah.
I did have one technical question though, and
We'll
we've got
try.
some others that we need to get to, but I'm taking advantage of my position here to ask one kind of technical question. And you chose an OMARS design. Why not a definitive screening design for this problem? Because again, you've got 3-level factors, and the number of runs would probably be a little smaller with a definitive screening design. And you could augment the DSD to add additional runs if you felt like that was necessary. But I just wondered if you thought about the DSD as a possibility.
Yeah, for sure. So what the answer that comes to my mind immediately, and Peili, you chime in too. One is that Peili and I took a short course on OMARS designs together, and so we were interested in exploring just as a new option. But second, and perhaps more importantly, is that the equipment setup is such so that we had 24 runs available to us, sort of like exactly 24. So we wanted to use all 24 exactly.
Got it. Yeah.
And so that a little bit smaller didn't help us out in that case. We wanted to use all of the equipment available to us. Does that sound about right, Peili, or you have other ideas?
Yes, and in addition to this, at that time when we all learn OMARS, I also feel interested about the way we pick the design because you can base on the correlation matrix and especially the powerful interaction term part. I'm not sure how to pick several 24 rounds study from different, but at that time, OMARS design kind of can use a tool to check different designs features. And then we can pick the one based on the information we want to apply. Yeah.
Good. Well, sort of a follow-up question then is, what about software? What software did you use to set up the design and do the analysis?
So analysis, ask us that in a few months because it's ongoing right now as we receive the data and are working through it. But the software, so Dr. Goose and his colleague Jose Nunez Arez have developed the effects software. That's like a web-based platform.
Okay.
And so that's what we used to design the experiment. I think there are a small catalog of OMARS designs available, like through some of their publications, just openly available.
Mm-hmm.
But yeah, it's new to both of us. So this effects software, EFFEX, it's like a web-based platform.
I've heard of this, but I don't know anything about it. So
yeah, so it's like I said, new to both of us. So we're not experts in the area, but that's related to the introduction of OMARS designs as
well.
I
have some professional curiosity in this because I have a feeling that the publisher for my experimental design book is going to start pushing me to do a new edition because the current edition is like 5 years old, and they start to get nervous when it's been that long. And a couple of things that I have thought about adding to the book. One of them is the OMARS design because I don't have anything on it in the book. I have a lot of material about DSDs, but nothing about OMARS designs. And so I may come back and talk to you more about software at some point on this. And the other thing I'm going to add is something that I don't know whether you're familiar with these designs that are used for a lot in software testing. They're fractional factorials, but you can't necessarily fit models with them, but you can see whether certain pairs or triplets of combinations of factors produce errors. And I'm trying. I teach in a school where we have a computer science program, and they have a lot of students in software engineering. And I'm trying to lure the software engineering students into taking design of experiments, and hardly any of them do. But they all have to do software testing.
Yeah, and the applications of DOE are wide-reaching, right? So,
they sure are. But anyway, I'm fascinated by this, and I probably will want to get you to give me a little bit more information about how the results come out. and the software. So keep me in the loop on that, because if you're up for it, I'd be love to have you come back and talk about the results and maybe about the software. That would be a
for sure.
really interesting topic, I think. And when I have to get pushed to think about revising the book, I may come back for even more help. Okay.
Yeah, we'll do our best. I know Peili and I are both excited because, you work on the design of these experiments and spend a lot of time, but then the actual execution also takes a long time. We're not
Yes.
really involved in the actual execution, but the scientists are hard at work in the lab, the analytical scientists conducting the analytical testing. And so it feels like we got all excited for this experiment, and then you just wait for a long time while they actually execute it.
Right.
So we're excited to see the results and what we can learn.
Do you run into problems during that part of the work, getting the experiment run the way you've designed it?
I certainly have examples of that in the past.
Yeah.
And in particular, Peili may I'm sure you have more experience with this than me, but in a similar experiment that I've been supporting recently, a common situation is that you might lose one or more runs from the experiment due to issues that happen during the experiment.
Yeah.
Perhaps unrelated to the experiment itself, but the equipment just fails, and it's just can be difficult. And so for the example that we gave today, sort of a 1-block 24-run experiment, we're like, please don't lose anything. We really want these 24 runs. But sometimes, like in another experiment that I'm supporting, we actually have 3 blocks of 24. And so after every sort of block is complete, we spend some time saying, okay, what concerns do we have? We have a plan for block 2. Do we need to repeat anything from the first block that failed? Kind of adjusting course along the way while we have that equipment available to us. Peili, do you want to add anything?
Well, one of the common problems that I've run into in complex experiments with multiple factors is that when you change from one run to the next run, somebody forgets to change one or more of the factor levels.
Sure, that can be,
or they don't change them correctly. And I've always suggested that you make somebody on that experimental team responsible for actually going around and touching all the knobs, to make sure that they're in the right place. And sometimes I've actually seen a couple of cases where, because they didn't reset one or more of the factors, that it caused the equipment to fail.
Sure.
Yeah. So it's but I think that's an important issue in these complex experiments. So Peili, you had something else to add, I think, too.
Yeah, we do sometimes may face a situation like the experiment did not run as planned. That's why we usually just tell scientists that if it's not run as they expect, recall what really happened. So if we run on the wrong condition, and usually it's one a big message, maybe just 1 run or 2 run, and just let them know when we do analysis, just let them know that it do have a shortage. Some problem happened to this analysis, but the data still should able to, it's up to how much problem happened, but usually still should able to get some information for them. And for sure, if it's a bigger problem, they basically do need to rerun whole study. Yeah.
Okay. We have some questions that have come in from our audience. Let's see if we can cycle through these. Can you see these questions?
I see maybe 1 or 2 in the chat. Is that where we're looking?
Well, I've actually got it projected on a screen that I can see it.
Okay. Then you read it to us, and
let me read it. I think that's what we typically do. Okay, so here's the question. It says, how can a power analysis be performed for an experiment involving biological replicates with multiple technical replicates? If I plan to design an experiment to test a drug that reduces protein X levels in cell culture with control, treatment 1, and treatment 2 groups monitored over several weeks, is it possible to use power analysis to determine the duration required to detect differences between the treatment groups and the controls? I'm not sure I have a really good answer for that. Maybe you have some, maybe the two of you have some thoughts on how to address that.
So long question. I'll try to answer. Might need you to repeat some of it. I mean, I think the short answer, or the first thing that comes to my mind, is that we do conduct power analyses, in order to appropriately size different
Right.
experiments. Obviously, you have to have some historical data available to you, for example, to estimate a standard deviation. You have to have some knowledge about what size effect, what size difference between your control and treatment groups you're interested in, and then define a confidence level, things like that. So certainly those calculations or those kinds of analyses are things that we would use to inform the design of different studies. There were some specific questions sort of or specific details given in the question that I didn't retain all of them. You can, Peili,
Yeah,
did you have any thoughts, or do you want to read it again?
well, it says to test a drug that reduces protein X levels in cell culture with control, treatment 1, and then treatment 2 groups monitored over several weeks. And I guess you have a control group, and then you have a test group, and it seems to me that if you have the information that you're talking about, some idea of effect size, even if you don't know much about the standard deviation, you can always think about the effect size relative to the standard deviation. Are you interested in a 2 sigma size effect, which is fairly small? Or are you interested in a 3 sigma size effect, which is fairly large? And I think you could approach it that way. Does that make sense to you?
Yeah, certainly. Yeah, and I mean, I just can confirm we do those types of analyses in terms of course, figuring out sort of the size of different designs. How that's the first question scientists often ask us. How many samples do I need to test?
All right.
And we have to say, wait, back up a minute. I can't just tell you 30 every time, right? We have to really think about this.
Yeah, I've had the same experience. Well, I hope that our attempted answer here was helpful. But if not, you can always email me and we can try again. So, okay. Question 2: could you please explain how non-clinical data is concluded at the end of the study that could have been conducted on the animals?
So Peili and I, neither of us work in drug safety with animal studies. That is another role that non-clinical statisticians have. So supporting the design and analysis of experiments of safety studies that include animals. There's a group at Pfizer that supports those types of trials. I don't have, Peili, do you have any experience with that group? Yeah, me neither. So we don't specialize in that area, but it is, a very interesting area. I know one thing, I know very little about this, so don't quiz me on it, but I know that one big topic in this area is the idea of virtual control groups. So, you'd have your control group and your treatment group to study the impact, does a new drug cause cancer in rats or something, which could be concerning, in humans. But trying to take advantage, kind of similar to the idea Peili talked about today, of compiling previous studies, so previous control animals, and can you borrow some of that data as a control sample, a control group for your current study? Since, the animals, like the rats, if it's a control group, they kind of should be the same, over time, because they're just there for a baseline. But anyway, that's a long way of saying I don't have experience in that area, but it is another area where statisticians contribute to the design of animal studies.
Well, I have to admit I'm sort of in the same boat with you. I don't have very much experience with animal studies. My only real experience with animal studies goes back into the dark ages of my career, before either of you were born, I'm sure. This is back when I was at Georgia Tech as a faculty member in the early mid 70s. And I was part of a group of faculty that did a lot of consulting work for Coca-Cola, the guys right across the street from Georgia Tech. And I got this urgent phone call from the guy who was the vice president of corporate engineering, who later went on to become the CEO of the company. So he was a very smart guy. And he said, we've got an explosive situation on our hands. Can you come over and talk to some of our flavor chemist people? Oh, okay. So I go over to talk to the flavor chemist, and they showed me this study that had just been recently published on how saccharin caused cancer in Canadian rats.
Mm.
And this is a disaster in the food and beverage industry, because saccharin was the sweetener that was used in diet beverages. Coke's diet product at the time, they only had one diet beverage, and it was Tab. And the sweetener was saccharin. And an all-out effort went from that point on to develop different artificial sweeteners. And I was lucky to work on that project. And the artificial sweetener that we developed turned out to be wildly successful. And it was the sweetener used in the new product they call Diet Coke, and which is a huge success.
Yeah, too bad you don't get royalties or something.
Yeah, well, I got paid a nice consulting fee, so I'm happy. But anyway, the long shot of this story was that after this study was published and sent these tremors throughout the food and beverage industry, the paper was shown to be based on flawed data.
Yeah.
They had fed these rats so much saccharin that on a human consumption basis, you would have to be eating pounds of it a day.
Yeah.
To have anything. Yeah. So I don't know whether the paper ended up being retracted or whether it was just politely ignored from that point on. But it was quite a tremor. And it did have a good outcome, because it led to a very successful new product for the Coca-Cola company.
Yeah.
Okay, here's another question. Hello, everyone. Presentation was nice and interactive. I was just checking how someone could be part of experimentation, design, and validation process. Is that kind of a question as to how you get into this?
I guess so. Yeah. I mean, so Peili and I are
how did you two girls get into this?
Yeah. Okay. Yeah, I can go first, Peili. You can. So I worked at a chemical company prior to Pfizer, and so after I got my statistics degree, I guess really I got into it while I was in graduate school. I did an internship at 3M in Minnesota and really sort of fell in love with applied statistics. So I knew I was looking kind of for that kind of job where I was sort of the intersection of science and statistics. So I worked at a chemical company for a few years, and then
that was Eastman Chemical, right?
Yep, worked at Eastman Chemical Company for 7 years, and then sort of right after the pandemic, made the switch and got a job at Pfizer. So doing similar work to my first internship, but in the pharmaceutical business now.
Great.
Peili, I think your story is a little different. How did you get involved in being a CMC statistician?
Actually, so my major was statistics for a long time, and I like statistical consulting. And my work before is more like a bioinformatics data. So I actually need to use R, do a lot of programming. And then after I graduated from University of Florida, then it's really just get a chance to meet this group and they told me their works about statistical consulting. And the scientists will have different type of question, and DOE just one of them. And we actually have different parts. So I kind of use R and then use the database idea from bioinformatics to working with this group about the DOE. Yeah.
Well, thank you both very much. I really appreciate this. This is the end of today's session, and I think we're going off air so that we terminate everything exactly at an hour. And thank you again. And I do want you to keep me informed on what's going on with this experiment, because I think the planning process of this is really novel, and I want you to think seriously about writing that up as a paper, because I think many people would benefit from this.
Thanks so much. Thanks for inviting us, and we'll definitely be in touch.
Yeah, and you can bet for sure I'm going to have you back to talk about the results.
Sounds good.
Okay, thanks a lot.
Bye.
Thank you.
Okay.